Today we are going to learn a major requirement of microbiology laboratory, that is cleaning, disinfection and sterilization. For your easiness, I have intended to describe the lesson in two sessions. So this is part 1. So in this session we have three objectives: to understand the impacts of contamination, to understand the scope of decontamination in healthcare setting, to understand the basic concepts of decontamination, that is cleaning, disinfection, sterilization, sanitization and antisepsis. To proceed with the lesson, let's understand what contamination is. Oops, this apple is spoiled. It is contaminated. But how? Maybe due to microbes. Next, you can see a blood agar plate which has been left in air for quite some time. Now, there is plenty of growth. It is contaminated, probably due to bacteria or fungi. So in this fashion, any sources like food, water, soil or air get contaminated involuntarily, either by chemically or microbiologically. Not only that, your culture media, instruments, even a laboratory or theatre space themselves are prone to get contaminated. One can define contamination based on various aspects, but in microbiology, it is non-intended or accidental introduction of microbes or their byproducts like toxins. So in any healthcare setting or in any microbiological work, contamination is an involuntary process and therefore we need to minimize. Otherwise, we cannot ensure biosafety, or at least we wouldn't be able to isolate the pure growth. Let's explore impacts of contamination. Haha! Except the historical contamination which led to discover penicillin in good old days, all others have brought negative impacts. Let's see those negative impacts of contamination, especially in healthcare settings. First is the environmental bioburden. This is all about microorganisms exist around us. They could be airborne, found in droplets, or rest on surfaces we touch involuntarily within our own premises. We can get contracted with them either directly or indirectly through three I's, that is inhalation, ingestion or incision. So we are prone to get exposed to them constantly and might get infected. Organisms living inside laboratories, ICUs, are constantly exposed to antibiotics as well. So they are more resistant strains than their wild counterparts. We identify them as multi-drug resistant organisms or extensive resistant organisms like Acinetobacter, Pseudomonas, MRSA, VRE or even mycobacterial species. So contamination accounts for occupational health hazards and hospital cross-contamination, more precisely healthcare associated infections. It is estimated that contamination contributes lavishly in HCI worldwide. Therefore, we diagnosticians are compelled to prevent or minimize contamination wherever possible, unless we have to pay more for the consequences. Let's turn to a microbiology laboratory. There the lab worker, equipment, specimen and environment are the key elements that are in high concern. We need to protect lab worker from getting infected, thereby preventing occupational health hazard. We have to make sure that his equipments and procedures are free of contamination, which directly affects for all the results he made at the end. One of his primary goals should be to protect specimens he received. If not all his effort, time and money invested for the analysis are in utter vain. He will have to take care of his environment as well, especially in waste disposal. Finally, he needs to comply with regulations and laboratory accreditation. So it is obvious that prevention or minimizing contamination in healthcare setting is a mandatory process and our primary obligation. Next, we dig out the scope of decontamination. When it comes to healthcare facility, it could be laboratory, ward, OPD, theatre, ICU or any other place where healthcare is provided. To understand more, let's watch the video on decontamination. There we saw surfaces, instruments and equipments to be decontaminated. Apart from that,
good hygienic practices to minimize decontamination. Next, let's have a close look at on things to be decontaminated in a microbiology laboratory, as it's our major concern here. Here also we see different types of instruments, equipment, surfaces and instances to be decontaminated. In microbiology laboratory, you can categorize these events as pre and post procedures or as disposable or reusable items. You would be able to categorize them more once you learn decontamination methods. Next we see how we prevent or minimize contamination in laboratory. It is achieved through decontamination processes and good laboratory practices. For that, identifying the source is important. Then we can simply apply decontamination as a corrective measure in all pre and post processes of laboratory work, followed by good laboratory practices like handling cultures, biosafety measures, wearing PPE, and so on and so forth. Decontamination consists with three arms: cleaning, disinfection, and sterilization. In fact, decontamination plays a pivotal role in infection control. Cleaning is the physical removal of gross contaminants. It is an essential and fundamental process in decontamination. Routine practice is either dry clean or wet clean by manual or with the aid of electrical appliances. For wet clean, water with or without detergent or enzymes like proteases can be used. It apparently reduces organic matter and microbial load. It invariably helps to increase the efficiency of subsequent decontamination processes like disinfection or sterilization. Let's recap and categorize things to be clean and decontaminated in microbiology laboratory. As you can see, those are critical and non-critical surfaces, instruments, and liquids. Here we have more questions. Does cleaning remove all the contaminants, organisms, or all the pathogens? Does cleaning ensure biosafety? Cleaning is usually along with a detergent or a disinfectant, and why is that? And finally, do we need to eliminate all the organisms at all instances? However, to cater the above requirements, efficacy of cleaning can be improved. Here we have to consider following factors: awareness of cleaner, method, frequency, surface and percentage of contamination, efficiency checking, and finally monitoring. When it comes to awareness of cleaner, he should be able to understand percentage of contamination and selection of method. Method, with or without detergent or disinfection. And if it is frequency, whether hourly, once daily, twice daily, weekly, or so on and so forth. There are critical surfaces and non-critical surfaces. Critical surfaces are benchtop, door handles, computer keyboards. Those are required to frequent decontamination. Efficiency checking. It is a bit controversial. Still, we have to satisfy with visual methods of checking cleanliness, because we don't have microbiologically accepted efficiency checking method. Monitoring is important. We have to have checklist and appoint an authorized person. Cleaning can be improved by detergents. They are surface active agents or surfactants, having lipid-soluble and water-soluble components in the same molecule. They disrupt lipid-containing cell membrane of microbes and cause to death. Apart from detergents, many chemicals are being utilized to control microorganisms. Bactericidals are those that kill microorganisms, and bacteriostatics are those which inhibit the growth of microorganisms. Usually, antibiotics are not used in disinfection purposes. In mechanical cleansing or sanitization, all the forms of organisms are not killed, but bring down to a safe level, especially in food safety. Antiseptics are non-antibiotic chemicals specially used to eliminate pathogenic organisms on skin or mucous membranes. Routinely used skin antiseptics are 70% alcohol or povidone iodine. Being laboratorians, it is our fundamental duty to maintain aseptic conditions in all our exercises within laboratory. Getting contamination-free environment, equipments, enhancers with good laboratory practices are important in maintaining proper decontamination. Disinfection is the process of elimination of pathogenic organisms on inanimate objects. More resistant forms of organisms like bacterial spores are not killed by disinfection. Routinely, chemical agents used for disinfection, and they are called disinfectants. Laboratory has wide variety of disinfectants. Aldehydes, phenols, chlorines, oxidizers are few of those categories. Example, 2% glutaraldehyde, 5% lysol, 1% hypochlorite, but those are not used on skin or living tissues. Only 70% alcohol and isopropanol are used for both skin antisepsis and surface disinfectants. More important fact is that those should be freshly prepared with appropriate concentration for optimal disinfection, and contact time is another important fact. Apart from these, there are many physical methods of disinfection like UV filtration as well. These disinfectants can be categorized into three levels based on their activity against microorganisms. They are low level, intermediate and high level disinfectants. You can increase the concentration of some low level disinfectants to make high level disinfectants. But none of these are sporicidal or sterilants. Sterilization is the process of freeing an article from all the living forms of microorganisms, including their spores. Sterilization can be achieved by physical, chemical or physico-chemical means. Major sterilizing agents are autoclaving, hot air oven, ethylene oxide gas, hydrogen peroxide, plasma gas and liquid chemicals. And it is the maximum safer level in decontamination. Finally, efficacy of decontamination is determined by four major factors. One will have to select an appropriate method, whether it is disinfection, sterilization or just sanitization, and whether heat method or chemical method depending on the requirement. Then the surface or article to be decontaminated, whether heat labile, critical surface or its percentage of contamination. Next is the resistance of organisms expected to be contaminants, whether they are mycobacteria, sporoformers or non-capsulated viruses. However, good laboratory practice is the baseline in all efforts of minimizing and prevention of contamination. With that, we finish the first session. In next session, we hope to discuss on sterilization and disinfection methods, their efficacies and limitations. Thank you.